Okay, so that's enough, and I guess at the end you can have a discussion on these things if you would like. I would like to go on to Pete, who's been the scientific committee chair, and I have to admit I've heard these things every 3 to 4 months for the last 3 or 4 years. I am very impressed with both these two, both the scientific committee thing that you're going to hear about, and also the education committee. Very impressive work, I must say.
Thank you, John. Okay. Scientific committee is responsible for vetting studies that are proposed. We have a format. We have a sort of form that you can submit if you have ideas or concepts, and that will be talked about, discussed openly, and then an adjudication made. But it's actually a fairly informal process, and then if approved, it gets an MSG number. Currently, we're at MSG 20, which is also the Mario study, which I'll explain in just a second. It won't take long. Okay. One of the activities that we do, and I kind of indicated this earlier today, was we advise companies who come to us with questions regarding best design for study. And so we have been involved in the last really 3 to 4 years now, since the last meeting they've had, with about 8 entities, and 9 including the CDC. And we have provided them advice. We've met either by Zoom or face-to-face, usually with a consulting committee, people with expertise in an area, et cetera, and sit down with the sponsor, whether that's a CDC or a company, an entity, a pharma or a diagnostic company, and go through, kind of our thoughts about how to do this fairly, transparently, making certain that a study is designed properly, with the understanding that we'll have a greater or lesser role once the study is rolled out. And so these are the types of things we've been involved in terms of our advisory activity. If you look at the studies that we have been involved in, and many of them are done now, not all of them are, in fact, these include studies that are not yet published. So this includes the individual studies that are not yet published, but have recently finished. Okay, now, so MSG 11 is it's the international counterpart of the CINCH study that Peter Williamson and Karen Marr completed and published a couple of years ago. That included 150 U.S. patients who were non-HIV positive. This study enrolled 103 patients, and unfortunately I don't have the distribution of patients, but the bulk of them came from Taiwan, Australia, India, Peru, West Europe, and there were a couple from West Canada, from the western provinces of Canada, the Vancouver area. 103 patients. We do have isolates on most of those individuals. Once we have them shipped to Sean Lockhart, once we get our agreement completed, they'll be logged, retyped, and you can, at that point, you can do what interests you with them in terms of susceptibilities and so forth. But what's captured here is outcomes, how the diagnosis was made, the basic kind of stuff you would want to know regarding these sorts of patients. How long were symptoms? Did they survive? How long did they receive treatment, et cetera? So haven't looked at these data, but it'll be interesting. And again, these are non-HIV included patients. The largest number of patients actually came from Taiwan. And Peter and I were talking earlier today about the large numbers of patients in Asia, especially China. So if you were to do an interventional study or repeat this, even if you couldn't get onto mainland China, then Taiwan is a very willing and very cooperative and really good partner. The people that will write this manuscript, I'm listed at the PI, but all I did was look at reports. I didn't enroll a single patient because that's not our job. But Sharon Chen and Dr. Chen in Taiwan will be writing this up once the analysis is done. MSG 14 was a prophylaxis survey. And this is headed up by John Badley, Tom Patterson, Oliver Cornelli, and others. And it was basically to look at what sort of antifungal prophylaxis patterns people at different institutions use. The focus here being primarily lung transplants and allogeneic stem cell transplants. I don't have the data from that, and I can't provide a up-to-date status report of that at this point. It was sponsored by Maine. That study's had its fits and starts. It's mainly a survey. Study 15 is a study that I mentioned earlier today. This is the most recent study that we've conducted that was sponsored by Maine generously, and it was the first endemic study done probably in over 20 years in the US, or close to 20 years. And it involved 88 patients we enrolled 88 patients in about 2 years, all done in the US with one exception. We had a site in Panama and 15 sites. About half the patients were histo, about a third blasto, and then the remainder were cocci and sporotrichosis patients. That study is being written. Now the analysis has been done. Andre Speck is going to be the lead author of that, and GR will be co-lead on that. MSG 16, there will be one paper. We were thinking of 3, but there'll be one paper that's generated from this. This is going to be the combination of the clinical, the PK, and the quality of life survey. All this was being captured, and we thought that perhaps as a PK safety study primarily, this would turn into 3 nice publications, but really the best story is told with one, and so Andre's agreed to write this up. The natural history of antifungal failure, MSG 16. It's kind of quasi-retrospective, some retrospective, some prospective, but it's basically individuals who fail antifungal therapy with candidemia. It's being run by Louise Ostrowski-Zeichner, the incoming president. 125 patients as a labor of love. Louise and his group have really sweated over this, worked very, very hard. The DRC is underway now to adjudicate the cases, and then once that's done, then that will be written up. There are 6 sites, I think, involved in that, 7 sites. No, 5 centers, excuse me. MSG 7 we just wrapped up. This is a really good example of something that starts out as kind of an epidemiology study, but really morphs into also a diagnostic and a treatment study and an outcome study. This is COVID-associated fungal infections, with the emphasis being aspergillus, but honestly, it's all fungal infections. And this came about, CDC spawned an idea. Tom and his group came to us and asked if we would be interested in pursuing this. We couldn't do it with the budget that was allotted. We got Astellas to chip in significantly, and we were able to recruit 7 sites in the end. These are all ICU patients who've been in the ICU to qualify for the study, you have to have COVID, and you have to have been in the ICU on a ventilator for 72 hours. That's it. From these sites, we were able to recruit 221 patients. The analysis is underway. The data have been cleaned. When this analysis is done, this paper will be written primarily by Hong Nguyen. Our group and her group enrolled the most patients. And she really had a lot to do with the design of this. MSG 18 is a concept. It's been sent as a proposal to NIH as a broad agency agreement. It was funded by an R34. This is, we talked about this briefly today. It's a study that could use a number of different study agents, but the focus is Candida auris and drug-resistant Candida. The rules for enrollment are a bit different than what we've seen in the past. I won't go through that in any detail now, but we thought the compound ATI 2307 was interesting. It was unique, had unique activity against all the Candida species, including those that are resistant. So it looked very attractive. And comparing it to an echinocandida and a standard of therapy. This study it'll sink or swim depending on whether NIH is all that interested in funding treatment trials for Candida due to these organisms. The drug specifically doesn't really matter. It's the design of the trial, and I think have designed it in a way that we can use other compounds. And David Bulwer even suggested maybe we could use this as a platform approach, where it's kind of a system approach to looking at multiple agents against Candida auris and these other organisms. So we'll wait. Once we know what the funding situation is with that, we will contact you. But presently we have sites in India, South Africa, and several sites throughout the U.S. We had originally recruited sites in Panama and Colombia as well, but we will see. All places that seem to be relatively rich with Candida auris and or Glabrata cruzii and other of the target organisms. 19 is a study that was done by Jeremy Walker, who's one of our brand new faculty. And it really looked at we noticed, and John and his group noticed, that we had 5 or 6 patients with crypto following COVID. And it seemed to be temporally related and connected, which it's kind of hard to make that connection. But we said, well, let's write this up. Jeremy presented it as an abstract at last year's IDSA. It got a lot of good commentary. And so Jeremy and I talked, and we turned it into an MSG study. He made a very simple case report form, was able to send out a blast kind of survey to sites, and without having to get IRB approval, without having really to pay sites anything, very quickly, we received 70 cases, 70 completed CRFs from international and US sites. So the problem was much bigger than what we had anticipated. We really thought we were going to be dealing with 15 to 25 cases. Cases continued to just roll in. And at the end of the day, he ended up with about 80 plus cases, but had to cut some because the data were just inadequate. This is being written up. And so that data has been analyzed, and we're just in the process we'll pass the manuscript to those who are involved just as quickly as we can. And the last one is MSG20, the Mario study that Nkechi talked about yesterday. Our role in this study, again, this is a large study. It'll enroll 200 plus patients. It is intense in terms of its monitoring. It involves at least 50 international sites. This is nothing that we can do. We're not a CRO. We could hire a CRO, but so can Synexus. So our role from the outset was to help design the study. And so we are responsible for the design, for better or for worse, with Synexus at the table. We will be responsible for the DRC that adjudicates cases. We'll be responsible for looking at the analysis, making sure that it's fair, and writing the manuscript. We'll also, once we've agreed to terms, also be helping to encourage sites, helping sites to recruit patients. So I think it's a good marriage, and I think we'll likely be doing trials that are more designed like this. Anyway, that's our current plate of current, active, and recently completed studies. that, I think, is the end of my presentation. I did want to say before I left here that we need to recognize John Perfect for, first of all, enduring for 4 years, because he had to stay longer than, he expected. 2 years is the term, but he graciously elected to stay on and continue to lead us and run the board meetings. And frankly, John, while not uniquely responsible for this meeting, was the prime mover behind the content of this meeting. It would look very different if I were in charge. It would have been less rich with really good scientific data. So if you've enjoyed the meeting and you want to thank someone, in addition to the staff out there and from Castle and Associates and our central unit staff, make sure you compliment John on this, because he's largely responsible for the content of this meeting. Thank you, John.